Alright, so in this lecture, let us talk about docking. Okay, and docking is a method of structure-based drug design. Docking is a method of structure-based drug design. And of all the methods that we have in computer-aided drug design, docking is the one that is most widely used. Okay, so docking is the most widely used method of drug design. Leave aside structure-based drug design. Okay, so it is a method, is an example of structure-based drug design. Okay. So let us talk about docking. Docking is a method of structure-based drug design and is widely used in virtual screening. So one method of virtual screening we have just seen in the previous lecture. We talked about pharmacophore-based drug design, right? 3D screening. Now another method of drug design for especially for virtual screening is dock. Okay, and docking is the most widely used method of computer-aided drug design, and it is more widely used than pharmacophore-based drug design for virtual screening as okay. Now, docking is nothing but finding a binding geometry between two interactive molecules of similar structure. Okay, so we have one receptor, and we know that a particular part of the receptor is the active site, and then what we'll do is we'll define that as the active site, and and then we'll make different ligands to bind to this active site. Okay, so before we use the ligand for binding, we should have different conformers of the ligand which have been generated. Okay, and the lowest energy conformers, as we had discussed in one of our previous lectures, it is always the lowest energy conformers which bind the best, right? But it is not compulsory because we know that both ligands and receptors are constantly in motion inside our body, right? So that means the ligand the lowest energy conformer might bind to the receptor, but it might change its conformation, either its own conformation or it might lead to a change in the conformation of the receptor. Okay, so that's the reason why we need to talk about flexible ligand, flexible receptor kind of docking. Okay, so what is docking? Docking is the finding the binding geometry between two interactive molecules which are of similar structure. So we have a receptor, its active site, and then we'll try to dock a ligand into the active site of the receptor. Now, since the ligand is complementary in shape to the receptor, and since it is complementary in charge with the active site of the receptor, this ligand is going to bind with a specific geometry to the receptor. And it is this binding geometry that we need to find out, and hence docking is used. Okay, so docking is nothing but finding a binding geometry between two interactive molecules of similar structure. And molecular docking is a key tool in structural molecular biology and computer assisted or computer-aided drug design. The goal of ligand protein docking is to predict the predominant binding mode of a ligand with a protein of known 3D structure. So here what is essential for us is we need to know the structure of the protein. It is absolutely compulsory, it is imperative that we know the structure of the receptor. Okay, those methods in which the structure of the receptor is known, but the structure of the ligand is not known. Okay, such methods are called as structure-based drug design, right? Receptor structure-based drug design. So docking is a method of structure-based drug design. So what are we trying to do? We are trying to find a binding geometry. And then we have talked about two methods of docking, right? One was ligand protein docking, and the other method was protein protein docking, right? So ligand protein docking, and we are talking about the small organic molecules which will go on to bind to the proteinaceous receptor, right? On the other hand, protein protein docking is nothing but one protein or a peptide binding to another protein or a peptide. And we know larger number of proteins and peptides are nowadays being approved as drugs, right? For example, monoclonal antibodies are nothing but protein. So protein protein docking is also peaking. Okay. Initially, in the earlier stages of molecular modeling, the history of molecular modeling, we used to talk only about ligand protein. But now we talk of peptide or protein docking as well. Okay. So here we are talking of ligand protein docking is to predict the goal is to predict the binding mode of the ligand with a protein of known 3D structure. Okay. Then all docking programs generate hypotheses about probable spatial positions of the ligands in the active site of the target molecule. So if we have a active site this way, okay, and then we use this docking software, what will it do? It will give us different poses. Maybe this is pose number one, maybe this is pose number two, maybe this is pose number three for the same ligand, right? So docking programs are going to generate hypotheses about the probable spatial positions of the ligand, spatial and a three-dimensional positions of the ligand in the active site of the target macromolecule. Now, what are the steps for docking? So, step one would be for each of the candidate molecules selected for docking, several ligand conformations will be usually searched against a limited number of receptor conformations. So usually what is done is the receptor structure is kept flexible, but only one conformation is used. Okay. So a receptor can be flexible. It can have this conformation, it can have this conformation, it can have some other conformation, right? But only one particular conformation is usually selected for the receptor. And different conformations of the ligands are selected. Okay. And then what we do is we try to dock all those different conformations into the active site of the ligand. So this leads to a combinatorial explosion in the number of possible ways of docking an individual molecule, each of which must be evaluated. Okay, so combinatorial chemistry we have already studied. So combinatorial chemistry is similar kind of chemical reaction happening with many different starting materials leading to generation of large number of products at the same time. Right. So similarly here also we have one fixed receptor structure, okay, which is conformationally flexible, but one particular conformation of the receptor is selected, and different conformations of the ligand are docked onto the active site of the protein. What will happen? We'll have many different combinations. So that is nothing but a combinatorial explosion of the number of possible ways of docking an individual molecule can have with the receptor, and each of these methods should be evaluated. Okay, it is essential that all of those conformations, how they are binding, that should be always checked. It is not that some conformation is binding weakly, and some other conformation is binding strongly. So we need to think only about the strong conformation. No, we have to think about all those different conformations and we have to evaluate them properly. Okay. Then step two is assessment of these hypotheses is carried out by using different types of scoring functions. Okay. So what kind of scoring functions we can have? We can have binding energy as the most widely used. Binding energy is the most widely used scoring function. Then we can have area of contacting surfaces. So for instance, a ligand can have hydrogen bonding, it can have hydrophobic interactions, it can have, other ionic type of interactions, other type of interactions, right? So all of those different types of interactions can also be used for scoring. But that scoring function which is most widely used is binding energy. Okay, so binding energy is the most widely used scoring function. So when we all use docking for our own molecules, whatever we have designed, or maybe known molecules and finding the docking with the particular receptor, then what we do is we kind of evaluate their binding energy as to what is happening with those molecules and how they are binding with the receptor. Okay, so what we'll have is different binding energies given out. For example, molecule number one is having a binding energy of, say, suppose minus 7.8. Okay, molecule number two has a binding energy of, say, suppose minus 1. Okay, molecule number three has a binding energy of minus 0. Uh, 0.25 maybe. Okay, molecule number four has a binding energy of plus 1. Okay, so we'll have different binding energies which are calculated by the different scoring functions, and then we will rank those ligands accordingly. Okay, so here what we need to see is what is the greatest negative value. See, when we talked about enthalpy and entropy, we said we talked about the Gibbs free energy gene, right? Delta G. So delta G it has to be negative in order for binding to happen. So larger the negative value, better would be the binding. So in this case, minus 7.8 is the largest negative value. So that means this is the best binding molecule. Minus 1 is better than minus 0.25. So this is the second best binding molecule. So third best binding molecule. And this is plus 1.2. So plus indicates that there is no binding at all, right? Plus indicates that although the molecule, the ligand may come nearer to the receptor's active site, it may not bind. It may just have few interactions and it will be repelled away. Okay. So plus value indicates that this molecule is not binding at all. All right. So that's the reason why we had studied delta G, Gibbs free energy change earlier, right? So minus 7.8 is the greatest negative value, and this is the best binding molecule. Whereas plus 1.2 is not at all a negative value, right? So here the binding is not happening. Delta G is positive, that means the binding is not happening. All right. Then step number three. Based on the scoring results, the compound is the best correspondence to the binding site structure and properties are selected. So in this case, we know that minus 7.8 is the best binding. So what we'll do, we'll take up this molecule and we'll take up this molecule further for hit modification, hit to lead, modification, and then lead optimization. Okay, so this is what is the importance of scoring results or scoring functions in docking. All right. And then the obvious advantage of this method, that is docking method, is its high rate of obtaining results and ability to test numerous compounds. So initially, when docking was started, when the researchers started using docking, the algorithms were very, very slow, and the results that were obtained were, they used to be very delayed. So once a docking algorithm was started, a software was started, it used to give results, per molecule. It used to take many, many hours. So if you have to dock, say, suppose 10,000 molecules or 1 billion molecules, it will take years together, right? But nowadays, what has happened is those docking algorithms have been made faster. So what they do is they analyze the docking of one particular conformation of a given ligand very quickly. So maybe in a matter of 1 minute or maybe even lesser, the docking scores would be predicted by the scoring function, right? So that means they will give us the ability to test numerous compounds. So now what we can do, we can test billions of molecules in a matter of few months or a few days, right? And what has also been seen is there is a high rate of obtaining results. So because the the docking algorithms are very precise, they are based on force field. Okay? So because they are very precise, they will give us more precise results as compared to the previously seen pharmacophore based drug design 3D screening. Okay. So we can screen billions of molecules and we can expect good results to come out of those screening. Okay. Then binding site identification. Now, this is very, very important when you talk of docking as well as any other method of structure-based drug design. Okay, so this is always the first step to be performed when we are doing structure-based drug design. So I talked to you all about X-ray crystallography and I had talked to you all about NMR, right? So NMR solution state method. Now these are the methods which are used to find out the structure of the protein. X-ray crystallography gives us the advantage that it can give us the information of the binding site. Okay. And then once we know the binding site, we can select that for docking studies. But NMR solution structures, most of the time, they do not have give us the information of the binding site. So in this case, we have to predict which would be the possible binding site. Okay. But binding site identification is always the first step in structure-based drug design. So if the structure of the target or sufficiently similar homologue is determined in the presence of a bound ligand, then the ligand should be observable in the structure, in which case location of the binding site is very, very easy. Okay, it becomes trivial. So here we are talking of X-ray crystallography or homology modeling. Okay. And then, however, there may be unoccupied allosteric binding sites that may be of interest. So we may, even as a part of our X-ray crystal structure, we may have the knowledge of a binding site wherein the intrinsic inhibitor, it is found bound in the bound state. But there may be other sites also which may be involved in allosteric binding, right? Allosteric binding sites, we all know the definition of allosteric binding, right? So if we have a protein, okay, and if we say suppose this is the active binding site, but there may be some other point near the binding site wherein the ligands can go and bind, and these points are called as allosteric binding sites. Okay, and such allosteric binding may lead to change in the conformation of the protein, which may lead to enhancement of the binding at the active site, or it may deplete the binding of the ligand at the binding site. So that means the unoccupied binding sites, allosteric binding sites, may also be of interest. In brief, binding site identification usually relies on identification of concave surfaces. Okay, so ideally, there are many softwares also which are available for doing this. Okay, like for example, there is a software called a site map in Schrodinger's software. Okay, so that can help us to predict which all sites would be possible binding sites on the given particular receptor. But usually those binding sites which are concave. Okay, so for instance, if this is a protein, okay, and this has a concave site here. Okay, so either this would be a binding site or this would be a binding site. So usually binding site identification is identification of concave surface from the protein that can accommodate a drug-sized molecule. So if we have a drug, it can accommodate either here or here, right? That also possesses appropriate hotspots, that is hydrophobic surfaces, hydrogen bonding sites, etc., that drive binding, ligand binding. So if this site has a larger number of hydrogen binding sites, hydrogen bond acceptor or donation sites, or it has larger number of hydrophobic sites, then this is the site that will be involved in binding over this site. Okay, so binding site identification is very important. Then scoring function, needless to say, scoring functions are absolutely important. Why? Because they help us in prediction of the ligand binding affinity. Okay, so in our previous lectures, when we were talking of delta G, we were talking about KD, right? We were talking about the dissociation constant, right? So that was an indication of by a ligand binding affinity. So similarly, scoring functions for docking, they help us to predict the ligand binding affinity. Now, this is very important step in SBDD. Yet the current methodologies that are used for prediction of ligand binding are indefinite and they are complex. Okay, so that means they are error prone. So although we may have a very good, docking between the receptor and its ligand, but still due to errors in the binding affinity determination, that can lead to error in the scoring function, and we may miss out on some of the most potent molecules. Okay, so that is a kind of drawback with docking. Now, structure-based drug design attempts to use the structure of the protein as the basis of designing new ligands, applying the principles of molecular recognition. So if we know what all parts are there in the binding site. So for instance, if the binding site is having a positively charged region, if it is having a region wherein there are large number of hydrophobic amino acids, then we can design our molecules accordingly. Okay, so we can do fragment based drug design. So we can have something like a COO minus group in our ligand, which will interact with the positively charged area of the receptor via ionic binding. And then we can have some, since these are hydrophobic regions, we can have some phenyl groups or, some other groups, alkyl groups maybe, which can have hydrophobic interactions with this part of the receptor. So this is what is mentioned over here. So we attempt to use the structure of the proteins as a basis of designing new ligands by applying the principles of molecular recognition. And then selective high affinity binding to the target is generally desirable since it leads to more efficacious drugs with fewer side effects. So if we have a ligand which is strongly binding, then definitely that ligand will, kind of be associated with the receptor for a larger amount of time. If the drug is associated with its own receptor, then that will definitely lead to fewer side effects. Right? So that is why selective and high affinity binding is important. Thus, one of the most important principles of designing or obtaining potential new ligands is to predict the binding affinity of certain ligands to its targets and use the predicted affinity as the criteria for selection. Okay, and we can also predict the affinity of the ligand to anti-targets. Anti-targets यानी those other targets जहाँ पे the ligand should not go and bind. The target is the receptor to which the ligand has to go and bind. We can also check it the affinity of the ligand for anti-targets, and in those different proteins or enzymes or receptors wherein the ligand should not go and bind. Okay, so with this kind of prediction, we can predict molecules which are more specific for the action that we are expecting out of those molecules. Most of the scoring functions are physics based, molecular mechanics force field based that estimate the energy of the pore within the binding site. And this is, for example, an equation, an additive equation, which talks about the various contributions to the binding. So, for instance, the entire energy for binding can be due to the Gibbs free energy in the, due to the solvents getting disturbed. Okay, the enthalpy change in the entropy change in the solvents, then the conformations, the internal rotations, the single bond rotations, different types of rotations and that are possible within the ligand with respect to the receptor. So all of them combined will give us a particular value, delta G bind, which is nothing but the binding affinity, right? So this is a additive equation. Additive equation, any all of these are combining together, right, to give us the value of the binding affinity. So let us talk about few key concepts in docking. So receptor is also called as the host. So receptor or host is the receiving molecule, most commonly a protein or other biopolymer within which the ligand will be binding. Then we have ligand or guest. रिसेप्टर बोलते हैं वहाँ पे ligand बोलते हैं जहाँ पे host बोलते हैं वहाँ पे guest बोलते हैं. Okay, so it is a complementary partner molecule which binds to the receptor, and ligands are most often small molecules but could be other biopolymers, like for instance we talked about protein protein docking, right? So in addition to ligand protein docking, we have protein protein docking as well. Then docking computational simulation of a candidate. Docking is nothing but the computational simulation of a candidate ligand binding to its receptor. So how a particular conformation of a ligand is going to bind to its receptor. So that process is called as docking. And binding mode is nothing but the orientation of the ligand receptor relative to the receptor, as well as to the conformation of the ligand and the receptor when bound to each other. Okay, so we have different binding modes. So binding mode यानी how a particular ligand can interact with the receptor. Okay, so those modes are called as binding modes. And pose is nothing but how a candidate has its binding mode. So one particular binding mode is called as one pose. Other particular binding mode would be referred to as second pose, and so on and so forth. Then scoring. Scoring is nothing but the process of evaluating a particular pose by counting the number of favorable intermolecular interactions such as hydrogen bonding, hydrophobic contacts, etc. So scoring will help us to rank the molecule, right? So the next concept is ranking. Ranking is process of classifying which ligands are most likely to interact favorably with a particular receptor based on the predicted free energy of binding. तो ये scoring functions के basis पे जो free energy would be predicted, right? On the basis of that predicted free energy, we'll be ranking those ligands. So in one of the examples that we saw, -7.8 would be that ligand which is binding the best. Right. तो -7 से लेके -8 के बीच में जो भी बाइंडिंग एनर्जी वाले ligands होंगे, they will be ranked higher as compared to maybe -1 के range वाले and बाकी के in that particular example, right? And then docking assessment is when we quantify the predictive capability of the docking protocol. So docking assessment is also called as validation. Okay, docking assessment is also called as validation. So validation is the most important step in docking. You know, softwares, most of the softwares are very easy to use, right? Even if we give the softwares to school children, they will also be able to use the software. Okay, but when we are using it, we have to use them from a point of view that we have to design newer molecules, or whatever is the objective or the subject of our study, that we need to accomplish, right? So we look at it from a different perspective. But whatever we are trying to do, unless and until we validate our docking protocols, our docking assessment, we are not going to get good results. Okay, so docking assessment or validation of the docking protocol is the most important step in any docking study. So these are illustrations. Okay, so this is a lock and key kind of illustration. So this is the ligand and this is its target, and it this cannot directly go and bind to the target. So what is happening, it is switching its orientation, its conformation, and then it is fitting into the target and forming the complex. So this is the concept of docking. Okay, and actually, if we have to see, so this is the receptor, and it will have a small space over here within which the molecule is going to dock. Okay, so as we can see here the molecule is docking. This we can see is the green colored part. Okay, now this is the actual illustration of how a ligand binds to the protein. Okay, so this is the entire protein, and this is where the ligand is bound. Okay, so that means this is the active site of the protein. Okay, so this is actually a image that has been taken. This is a beta 2 adrenergic receptor, and it is a GPCR, G protein coupled receptor. Okay, and into this we have this small green colored ligand which has been docked. Sorry. Then what are the different docking approaches? So the first docking approach is finding out the shape complementarity, and the other, obviously, needless to say, we also need to check the charge complementarity. Okay, and then we do the simulation. Simulation, yani the molecule would be placed within the active site of the receptor, and that is the binding site that we have defined, and then it will be simulated. Yani ki different conformations of the molecule would be generated inside the active site, and you know the best conformation will be code, and it will be given out for ranking, and then we'll be able to see that in the form of the ranks. Okay, the output file of the docking protocol. So this is the docking protocol. So we have, one is the search algorithm, the other one is the scoring function algorithm. So search algorithm क्या करेगा? It will either undergo systematic or molecular dynamics or local feature matching or genetic algorithm type of approach it will take, and it will lead to generation of different conformations of the ligand within the binding site. Okay, and then we have the scoring function algorithm. So it will use the force fields or empirical data or knowledge based function for scoring, and the different conformations that are produced over here will be scored. Okay, and then they will be given out for docking assessment. Okay, so docking assessment में what all we need to check? We need to check the docking accuracy, we need to check the enrichment factor, and we need to check the prospective pharmacological validation. Validation, as I had mentioned, is the most important step. Okay, and validation can be done. See, here also I mentioned validation of docking is the most important step. So if we have to validate, if we want to have good results out of a docking study, then definitely we have to validate our studies. Okay, docking study. So what does validation include? For example, if we have a native ligand, intrinsic ligand in the X-ray crystal structure of the protein, what we can do is we can extract that particular X-ray crystal structure of a native ligand, and then we can dock it back using our own protocol. Okay, and then we'll see how the ligand is binding. For example, let me show it using an illustration. So this is a protein. Okay, and we have a ligand which is bound to this protein in this particular conformation. Okay, so this is the native ligand, X-ray crystal structure. What we'll do is we'll separate this native ligand from the X-ray crystal structure, and then we'll try to dock it onto the same protein. So we'll see how our docking protocol is giving out the pose for this particular native ligand. So if it gives out the same way, then that is good. Then our protocol is validated. But what if it gives out something like this? Okay, look, the molecule is almost like a single standing line, right? But what if the docking protocol gives it like this? So that means this is error. So whatever predictions we will have out of this docking protocol will give us erroneous result. Okay, we have to refine our docking protocol to such an extent that the molecule should be the intrinsic ligand should be now bound in the original form, standing line form. So if this is happening, then whatever predictions we do out of this docking protocol would be good. This is also called as cross validation. Okay, cross validation of the docking protocol. All right. So this is a docking approach, a validation approach. That okay. There can be other methods of validation also. For animal solution structures, we do not have native ligand as a part of the receptor structure. So in that case, we can resort to the binding affinities of known ligand. Okay, so we can have known ligands which are known for that receptor. Then we can dock them, we can rank them, and we can try and find out as to whether the ranking is correlated. Okay, so if we have a relative mean RMSD, okay, relative what is this? Root mean square deviation. Okay, so root mean square deviation, if it is nearer to zero between the native conformation of the ligand and the original conformation of the native ligand and the docked conformation, so that will be good. So the larger the value of RMSD, the worse would be the docking protocol. The better the value of the RMSD, the more nearer it is to zero, better would be the docking protocol. Okay, so validation of docking is the most important. Okay, and we have also talked about ligand flexibility and receptor flexibility in our previous lecture. Let us just quickly revise it. So when a ligand goes to bind to a receptor, it cannot afford to remain rigid. Okay, or on the other hand, the receptor also cannot afford to remain rigid. Okay, we know that inside our body, both the receptors, all the receptors are in a dynamic state of motion. And the ligands also, we know they have C single bond C, right? So they will tend to be flexible in nature. So that means the receptor is flexible, the ligand is also flexible. Hence, we need to incorporate ligand and receptor flexibility into our docking protocol. Okay. So if we keep the ligand rigid or the receptor rigid, then that is not going to serve our purpose. Okay. So docking protocol should almost always be having ligand flexibility and receptor flexibility associated with it. Okay. We cannot just have a rigid receptor because we know inside our body receptors are constantly moving. We cannot have ligand, ligands that are rigid. Otherwise, they won't bind at all. Okay, because most of the times we know that the binding sites are concave pockets. As a receptor, it is not bending at all. It will not be able to move, right? So it has to bend and bind to the receptor. So that is what is ligand flexibility and receptor flexibility. Okay, applications of docking, binding interaction between small molecule ligand and enzyme protein may result in activation or inhibition of the enzyme. If the protein is a receptor, ligand binding may result into agonism or antagonism. So one of the drawbacks of docking is we cannot predict from docking, whether the given ligand is going to be an agonist or antagonist. We just can predict whether it is going to bind to the receptor or okay. Whether it will be an agonist or antagonist will be found only via in vivo studies. Then, docking is most commonly used in the field of drug design. Most drugs are small organic molecules, and docking may be applied for hit identification and for lead optimization. So, docking is used in both of these steps. in hit identification as well, in hit to lead optimization as well, and lead optimization as well. So all the steps of computer-aided drug design we can use talking. And examples of successful applications we have the structure of an approved drug called as dorzolamide, which is a carbonic anhydrase inhibitor, यानी कि diuretic. Okay, it was approved in 1995 and it was discovered on the basis of it was discovered this drug dorzolamide was discovered on the basis of talking. Okay, then we have many other examples of successful application of docking, but this particular example I've taken, this is imatinib. Imatinib is actually a monoclonal antibody that was found out via protein-protein docking. Okay, so dorzolamide was a ligand that was found out via ligand-protein docking, and imatinib was discovered or rather designed using protein-protein docking. Okay, so both successful applications we have, right? And additional examples, we have so many examples. FOX2 inhibitors, cimetidine, enfibertide, zolpidem, zolpiclone, and raltegravir. Then we have selective serotonin reuptake inhibitors, then we have zanamivir, which is an antiviral drug, right? Many atypical of antipsychotics. Many of them have been discovered using topic. Okay. However, this approach is unable to discover compounds with high activity. So this is actually a drawback. Okay, so the researcher should not expect to find any hit with dissociation constant better than 10 raised to minus 4. Okay, and the efficiency of ligand binding with the target is estimated by the value of virtual binding. That is the drawback. Okay, so very high affinity molecules cannot be found, but reasonably, drug discovery में docking has been very widely used and it has been very, very successfully used. Another major problem with not only docking but with all other methods of structure-based drug design is the inability to predict the real binding energy and to convert it to its calculated value. That is the experimentally determined parameters like KD, IC50, KI, etc. etc. So what we have is just the docking score. We cannot go directly convert those docking scores into these values, KD, IC50, KI, etc. Okay, so that is the other drawback of docking. All right. So with that, we have finished off docking, and in the coming lecture, we'll talk about QACR, which is another method of ligand-based drug design, quantitative structure activity relationship. Okay.